thank you very much for being a podcast to Robert's Universe.
Well, thank you for inviting me.
Yes. So we at Robert's Universe are interviewing people that have great interests in Korea or food, and in particular in pets. And today we have ANDREA Gonzalez. So on this show, could you please introduce yourself to the audience?
Sure. My name is ANDREA Gonzalez.
Yeah.
I'm from Portage, Michigan, and I work as a professor at Michigan State University, but have worked in the pharma field for 30 years before I decided to retire and start my new career as a professor.
Oh, so you started when you were 10 years old.
Yeah, I was in kindergarten when I graduated.
Oh, so well, now you're starting your new career as a professor at Michigan State University. So could you explain what you do at Michigan State?
Sure. Yes. I run the drug discovery program, and I'm also a professor in the Pharmacology and Toxicology group. So I'm hoping to help professors, who've done a lot of work in their research area for 10, 20 years, and they think they're ready to develop a therapeutic toward their favorite target, to help them do that successfully.
Okay. So can you explain what pharmacology is and toxicology is? Because, you and I have been in the industry for so long and we know that word, but I do, once in a while meet my friends and to tell them that, about pharmacology and toxicology, and they kind of get lost. So if you had to explain it to a 20-year-old, how would you explain it?
I would, and maybe we might have to edit this because
that's okay.
I'm not doing the correct definition as a professor. But pharmacology, I usually think of that as, understanding how a therapeutic or a drug, a therapeutic drug works.
Sure.
So it's all of the work that you do to understand, how it's working in the body, how it's affecting the pathways that might be involved in disease. Mm-hmm. Toxicology is trying to understand if there are any safety risks associated with a therapeutic. But there's all kinds of toxicology fields. There's environmental toxicology.
I didn't thought about that. Okay.
Yeah. Yeah.
Oh.
So there's all kinds of different types of toxicology. I'm thinking of it more from the perspective of a therapeutic and trying to make sure a therapeutic is safe.
Were your parents anybody in science or in pharmacology or toxicology? Like, how did you?
No, my parents were not scientists. My mom was a math teacher and my dad worked at the paper mill in town. So
a paper mill?
Yeah.
Like where they made paper? Yep.
So they made a lot of cartons for the cereal companies that were just, right outside of town in Battle Creek, Michigan. So
really?
Yeah. So it was the Brown Paper Company, the James River, Fort James, had a bunch of different names. So it's still there. Okay.
I always saw these recycle labels on these boxes. So do you know majority of those boxes are recycled? Is that?
They were. I think their paper plant was one that used recycled material. So the Kellogg Company was a big customer. So they did most of the cereal boxes for the Kellogg Company. So yeah, and that was pretty innovative back then when they were making a lot of those boxes because it was based on recycled paper.
And your mother was a math a math teacher.
Math teacher, yep.
Okay.
In high school, so we all had to be good at math.
Oh, and how then did you decide not to be an engineer, but eventually pursue a career in science?
I've always liked biology, so I just like to understand how life worked, whether it was in a cell or, parts of our body. I've always been interested in the life sciences.
Wow. I was always interested in playing games and, obviously we had different brains. Okay. And so you knew this early on?
I think so. I had a really good biology teacher in ninth grade. Actually, in eighth grade too. So we had really good biology teachers. So I grew up in Portage, Michigan, and in Kalamazoo, Michigan, there was the Upjohn Company. So that was a pharmaceutical company. So there were a lot of scientists in the area, and they had high expectations for science classes to be very good. So I think I benefited from that.
Did I tell you that my father had a joint venture with Upjohn back in the 70s and 80s?
Oh, so yeah, I think you didn't. So we have a connection there.
Yeah, we do. And that's why I told you I spent a summer back in 96 in Kalamazoo, Michigan, where I was staying at the dorms of Western Michigan University, I think, right? Yeah. So, okay. And from there, you then decided, once you decided toxicology and pharmacology, what was your first job?
I was a taste tester at the Kellogg Company.
You were?
Yes.
No way.
Yeah. So
really?
Back then, let's see, I graduated from college. This was before I went to get my PhD in toxicology. But so when I graduated from college.
Sorry, I can't stop laughing. A taste? What do you do? You're like...
I am a foodie, see?
I know. So do you like taste foods or?
Yeah. So at the Kellogg Company, they'd have test kitchens. And so they would, try to make a lot of different recipes with their cereal. So it could be like cornflake breading on chicken, or it could be Chex Mix, different flavors of Chex Mix. So they'd make these recipes in the test kitchen, and someone would have to taste them and, make comments and, write, is it too dry? Does it not taste good? Does it taste really good? So I had to do that. So I had to taste all of their recipes.
Interesting. Free food.
Yep. And I would eat the whole sample.
Well, isn't it like wine when you're tasting wine, you're just drinking and you're supposed to spit it out? Yeah. Isn't food tasting similar to that, or no?
No, I don't think so.
Okay.
But anyway, so I did, that was my first job. And then, then I did get hired part-time for a role, at the Upjohn Company. So I was a screener looking for inhibitors to reverse transcriptase, which is an enzyme in the HIV, the virus that causes AIDS.
Oh, okay.
So yes, I screened and found an inhibitor that worked really well that eventually was developed into a therapeutic called Rescriptor. So I think it's still used in some of the cocktails, but now they have a lot better drugs that are out there. No,
but you were one of the first, pioneers of HIV vaccine or medications, I guess. Yeah.
So Upjohn did have, had one of the first therapeutics out because everything was being fast tracked back then. It was back in the early 1990s.
Mm-hmm.
And so, it was an epidemic, and it was pretty scary. We're still learning what it was and how it was spread. And so there was a lot of money to look for therapeutics and to fast track those. So
I think that's a very interesting comment. I think it goes to just people are scared when they don't understand what's happening, right? It's like COVID. I think when first COVID went around, everybody thought it was the Black Plague. Yeah. Everyone was scared. When you coughed, like you wanted to be a mile away from everybody. Now, everybody understands what COVID is, and we just brush it off. But I think that might have been true back in the 80s too about HIV. We just didn't know how to manage it. There wasn't any treatments, but I don't think that people are as scared as they used to be, right? Right.
Yeah. And you have to always give time for the scientists in the medical profession to understand what's going on, and because that information may change and evolve over time. And so there's a little, sometimes there's not as much patience for that and as much anymore as there used to be, because the more you learn, your advice may update and change a little bit. So
we live in the internet world. We want instant gratification, instant information, instant results. Science doesn't work that way.
Yeah,
right?
Exactly.
So from Upjohn, then Parke-Davis, I think.
Then I went to grad school.
Okay.
And when I graduated, I came back up to Parke-Davis. I went to graduate school in North Carolina, Chapel Hill.
Oh, okay.
So loved it down there. The weather was great. And, but yes, I ended up getting a postdoc. I wanted to work in cancer research because that's what I was doing a lot of my work in as a grad student. And so I was offered an opportunity to do a postdoc at Parke-Davis in the cancer research group.
Okay. And so from Parke-Davis, you worked on cancer research. Was there another drug that you've developed at Parke-Davis that's used today as well?
Yes, actually, the cancer group was very experienced in developing kinase inhibitors.
Oh,
So
okay.
I worked as a postdoc there, did a little stint in toxicology, then came back to the cancer research group as a pharmacologist. So we were working on inhibitors to CDK4 and 6. Those are cell cycle control proteins. And so those were being developed for breast cancer.
Oh.
And so that inhibitor actually made it to the market. Ibrance. I did a little bit of work to understand, how it worked, and was the initial toxicologist on that program.
Mm-hmm.
And then I led a program that was the second generation EGF receptor family inhibitor. And so that one also made it to the market. I think it's called Visimpro. And it's used in for lung cancer treatment with patients that have a certain type of mutation in the EGF receptor. So not as big of a market, but still meets an unmet need for certain patients.
And, actually, this comes back to a conversation I had with some people who are not from the healthcare or pharmaceutical space, but a lot of people are focused on efficacy, how a drug works, right? And people say, oh, the FDA approved this, so this must be efficacious. But the FDA's role, of course, is important in the sense to find out if a drug is efficacious. But their main priority is to make sure that it's safe. Would you agree? I guess that I remember that's their main goal. Yeah,
I mean, both. I would say when you initially get out into the field, they're mostly concerned that it's safe, because they don't want to bring anything out into the patient population that is going to be harmful. And we learned a lot about that this week.
Sure.
And so, your initial goal will be to be safe because you want to give them the opportunity to evaluate efficacy. But to get approved, you're going to definitely need to show efficacy and in some cases differentiation from the standard of care. So,
but it has to be not toxic. That's the most important thing first, right? Yeah. Like you're there to treat, but it shouldn't be toxic, right?
Right.
Shouldn't make things worse. It should make things safe and good.
Right. Ideally, you want a very safe and effective therapeutic, but there's always risks to everything. So.
Okay. Well, you and I met not because of human health, but we met through the pet animal health industry. So, where did you go? How did you get into the pet industry? Was it after your work at Parke-Davis, or can you explain how you eventually got into the animal health space? Yep.
So Parke-Davis eventually became Pfizer. We were hostily taken over by Pfizer.
Oh, you're a Parke-Davis person.
Yeah.
Huh? Okay. Yes.
No, it was interesting. I didn't understand the difference between hostile takeover.
Don't worry, I'll edit
and that.
It out.
Yeah. So.
I'll edit that out. Yes.
I think that's what it was actually characterized as. But anyway, so eventually Parke-Davis was taken over by Pfizer and bought out by Pfizer. And they eventually shut the site down in Ann Arbor, Michigan. That's where the Parke-Davis site was. And then I had to make a decision of where I was going to move. So they had sites all over the U.S. Mm-hmm. And since I was from Michigan, I wanted to stay, in the Midwest. So I was mainly focusing on maybe St. Louis or Kalamazoo. Mm-hmm. And Kalamazoo would have meant moving to the animal health side of the company. And I had been working with them a little bit because a lot of times they would come over to the Ann Arbor site and ask to leverage compounds that we were working on to see if they would work in some of their animal models. So I really liked the people there, and my family, my parents were there. So I figured I would give that a try. And it was my first week. I remember listening in on a teat sealant project, and I thought I made the biggest mistake in my life going to the animal health side of things, because it was focused a lot on livestock, approaches.
Oh, okay.
And,
so you're doing human health all this time.
Yeah. And then I come into animal health, and there's all kinds of different projects. But eventually I found my niche in the companion animal space. So.
Okay. But you first started in cows and pigs and chicken
and every species, we were trying.
Aquaculture.
I don't know if they had aquaculture quite yet.
Okay.
But over time they did acquire
I
companies in that space.
Okay. And I always like to introduce you to other people as the inventor of Cytopoint and Apoquel. And a co-inventor, I guess, right?
Yes, there's many inventors that go on the patents for sure. So
could you explain to the audience what those two drugs are and how they help dogs today?
Sure. So Apoquel is a kinase inhibitor. It's a Janus kinase 1 selective inhibitor. And so it's an inhibitor that will basically reduce the effects of certain mediators that have kind of gone awry in the disease state. The animals tend to, that we're treating, are animals that have atopic dermatitis. So they are very itchy. They have inflammatory skin disease. And so a lot of the mediators that cause inflammation and itch will activate those JAK pathways, and Apoquel can inhibit that and block some of those mediators that cause those clinical signs. So usually most owners find it very effective. I actually have used Apoquel many times on my pets, and
you're not biased, right?
Yeah. I use Cytopoint the most, because I'm one that will forget to give the therapeutic to my animal. Okay. And then she gets itchy again and I feel bad. So I tend to use Cytopoint, which is a longer acting, and it's an injectable that works for about a month. So, okay. And that just targets one of the cytokines that Apoquel will inhibit, IL-31.
So if I had to explain that to somebody who's not, very familiar with science, there are two drugs. One is, they're both for dogs that have itchy skin, and one is oral, like you have to eat it, and the other one is an injection. Mm-hmm. And it basically, after a while, the dog, when they have bad skin, their skin gets back to normal.
Improves, yeah.
And it improves, and the dog itches less, which is also important for the owner too, because I've heard that owners, get stressed out the next day, or they couldn't sleep because the dogs kept on, crying, or were just bothered by, their skin, right?
Yep.
So it works that way.
Yep.
Would you agree?
Yes, I would agree. It's a very nice way to explain it.
Oh, well, I sometimes, that's not my job. Have you gotten letters from people? Have you heard from friends who know who you are and who know about Apoquel and Cytopoint, and have they shared some of their experience with you?
Yeah, actually, some people will share pictures. So I have someone at work whose dog's paws were really red, and then after Apoquel, the fur was nice and white, and the skin was cleared up. Yep, I'll get a lot of those types of pictures. I'll have, someone like, oh my gosh, I gave my dog Apoquel and they stopped itching within an hour or two, and they just went to sleep. So that's what I experienced with my dog. So I love hearing that. Because it's one thing to, do it in your experimental studies. It's another thing to see it work on your own dog, because you're knowing how much they're itching and scratching. So I hear that. So my financial advisor is so excited that his dog is on Apoquel.
Is he excited because his dog's on Apoquel, or is because your wealth is increasing, so he has more business?
I don't know if my wealth really increased much with Apoquel, but yes, he is happy to help me with building wealth, I would say.
I see. Okay. Wow. And you've also worked, not only with dogs, but I understand cats as well.
Yep. Yeah, we would do work on cats, and they also get skin disease. That one's a little bit trickier to solve because cats are not dogs, and things that work in dogs don't always work in cats. So,
yeah, I've had a similar conversation where humans are not dogs, and dogs are not cats, and cats are not humans. But unfortunately, right now, because there aren't as many treatment options for cats, that unfortunately sometimes I guess vets rely on human drugs, or or people make drugs based on human science, right, to make cat drugs. But I think that's changing. Would you agree?
Yes, I think we are understanding that each species is its own unique self, and so diseases can have similarities, but also uniquenesses. So I enjoy exploring those, differences and trying to understand those differences. And I think they're appreciating that the vet species deserves its own therapeutic that's designed specifically for them, that meets their needs. Because sometimes a formulation that works in dogs is not the ideal formulation for cats, vice versa. There's a lot of ways that you can tailor make a therapeutic that works better for a certain species over another.
I'm smiling because you told me your first job was a taste tester. Mm-hmm. Did you taste test the dog drug by any chance?
No, if I did, I'd have to put a report in for pharmacovigilance report.
An adverse event. Yeah. Because I mean, it's really difficult to medicate dogs and cats, right, if it doesn't taste good.
Right.
So did you have any difficulties making Apoquel taste good?
Yeah, so we could tell Apoquel was bitter from the beginning because our laboratory studies dogs, that were taking the medicine, we'd, find it along the cages sometimes, or, they wouldn't want to take it. So right away we did develop a chewable and palatable formulation for them, mainly a palatable one. I don't know how chewy it is. It is kind of soft, but so we worked on that right away to try to mask that taste. So, and that's Apoquel chewables out there now too. So.
Yeah. So now your dogs all the dogs are healthy. You take them out to the park. You do the picnic. I'm sure they're having a good time outside on the summers. Is that correct? Or?
Yeah. Well, I had 3 dogs. All of them had been on Apoquel at one time or another. 2 of mine have passed away. I just have 1 doggy left. So.
Oh, okay.
So,
but you still take them out, and then we were just talking about you having a boat in the lake, and you would have a barbecue, you would have friends over, right? Yep. Potluck when you do picnics?
Usually people bring a lot of food over for sure.
Okay. And since I'm from Korea, does anybody bring Korean food over, or? I
don't think so. No, but I would welcome any Korean food at the house.
I see.
What's your favorite dish? I should probably ask you.
Well, I have many favorite dishes, but what I realize that most non-Koreans like is a dish called japchae. And my friend explained it very well and called it Korean spaghetti.
Oh, okay.
It's a noodle dish. It's actually, the noodles are made with yam. It's with sesame oil, spinach, and meat, and it's mixed. I've had nobody complain to me that they did not like it. So I would say that if you ever want to try Korean food, you should try japchae.
Okay.
But more importantly, if I'm ever in Michigan, I'll definitely find a Korean restaurant and take you out.
Okay.
But what are some of the foods that you are very good at cooking, if I may ask? We were talking about that in the car.
So I think I went through 3. Okay. So I have a pot roast recipe. I like to make tacos. Corn tortillas are the best. Makes them the yummiest. Let's see. I have some pasta dishes, carbonara. I like to make breakfast foods too, like breakfast scrambles, that kind of
stuff. Do you wake up early?
No.
Okay.
No, I'm not I don't wake up super late, but I'm not going to get up at like 5 a.m. or 6 a.m. if I don't have to.
Mm-hmm. But you like steak and eggs kind of thing.
Yeah. Bacon. I like bacon and a great steak. So.
And you said that you're second or third generation Mexican?
Yeah, my grandmother was born in Mexico.
Okay.
But yes, my dad was born in the U.S. and so was I.
And so would it be wrong to assume that Mexican food is a very popular dish amongst your house?
Yeah, we love Mexican food. So I have a lot of aunts that were in the food service industry, throughout Kalamazoo. Some of them cooked at the hospitals. Some of them cooked at the university nearby, Western Michigan University. So we have a lot of cooks in the family. So they all compare their Mexican rice dishes, I guess would be the one.
Who's your favorite?
Well, it used to be our Aunt Elvita. She had the best.
Hi, Aunt Elvita.
Yeah,
hope you're watching the podcast.
She passed away many years ago. So everyone's been trying to replicate that recipe. So I think we all agree. One of my dad's cousins, Diana, who used to run, the food service, or at least some of the food service places, restaurants, or cafeterias at Western Michigan. She has the best rice.
Oh, okay. Well, I do look forward to trying that out one day if I ever in Michigan.
Yes, it's very good.
And
everyone
then I need to take you out to Korean food when I go there. I want to end the podcast with one question. If somebody was interested in entering the animal health space and they came to you and said like, oh, I too want to make, because pets are being humanized, right? I mean, everybody's having less kids and having more cats and dogs. So I only think this market is growing. There's going to be a lot more needs. And what if somebody said, like, I want to develop medicine or treatments for dogs and cats? What kind of advice would you give those people?
I would say that's a great area to be working in.
Yes.
I think there's a lot of unmet needs still waiting to be found and developed. So I think what I would say is, really try to understand the disease. Don't just try to leverage something from human health. Really try to understand the disease and, bring forward something that's really novel, because that clearly, if you understand the disease and what is the right treatment to bring forward, and it's novel, we've already seen, with Apoquel and Cytopoint, how that can stand the test of time and how people have just really valued those therapeutics. So I think there's a lot of areas still left in both dogs and cats and horses, even we consider horses companion animals. So there's a lot of people who would love to see treatments for laminitis, things like that. I think there's still a lot of opportunity to develop therapeutics in the companion animal space.
Is there such a thing as a area, a subject matter that I need to focus on? Biology, chemistry, math?
Obviously I'm biased towards biology and understanding disease pathology. So physiology is a good subject to have background in. Veterinarians know the anatomy and physiology quite well of the different species because they are different. So they're always valuable to have insights from. So those would be great skills to have to help.
Yep. I will make sure to tell that to my kids because, I'm an econ major and I'm trying to build this animal health company. And I have to say that lacking some biology, chemistry is very challenging. But, thanks for working with people like you and learning from people like you. I'm enjoying this journey, but I thank you for making time and sharing your personal experience and sharing that with my audience. And
Well, thank you for having me.
yes, and I look forward to many other perhaps novel medicines that come out of your lab.
I hope so. I hope there's a few more good ideas left up here.
Okay. Andrea, thank you very much.
Thank you.
Okay.